In this video, we're going to talk about genomics as it relates to cancer. But first, let's talk about how cancer works. So a tumor is essentially a growth of cells that shouldn't be there. So maybe it's just a little clump of cells, something like this. And these cells, they develop from normal cells that have somehow been damaged. And the tumors, there are two types. So on the one hand, you can have a benign tumor, and that means it's basically harmless. Or you can also have what's called a malignant tumor, and those are the cancerous ones, so they're bad. And only malignant tumors are cancerous because they have invasive potential, which means that they can spread throughout the body and cause a lot of harm. So we said that tumors result from a damaged cell, but what does that really mean? So if we go back to our picture of DNA, our double strand, right, and we have these nucleotides. Maybe those are, A's and these are the T's, and then we've got our C's and our G's. But maybe one of these is incorrect. So maybe instead of this being a CG pair, maybe this one right here, instead of being C, it was actually supposed to be a T. Right? So we have a mistake in our DNA. And we'll call that, that's one of our SNPs, a single nucleotide polymorphism. And so it doesn't seem like a big deal since we have 3 billion of these base pairs, but maybe when this gets translated into an amino acid, so right here, this could be our amino acid. Maybe this is, or this is a string of amino acids, and maybe this is the one in particular that is incorrect because of that DNA, right? So now that's our amino acid, and our amino acid is also incorrect, right? And if our amino acid is incorrect, then maybe our protein is deformed. So maybe our protein looks like this right here, but it was actually supposed to have a different shape, maybe something like this. And these are just hypothetical shapes, but if it is a different shape, it won't be able to do the same function, perhaps, that it was supposed to do. So maybe this particular protein is used in a signaling pathway. So for example, if the cell needs extra food or something like that, you need a signaling pathway so the cell can signal to cells around it and the body that it needs some nutrients. Signaling pathways are involved in all sorts of things, including cell division. So maybe this one's involved in cell division, which tells a cell when to divide, when to replicate. Then since the protein is deformed, the signaling pathway is damaged, and as a result, cell division is out of control. Okay, so now we have cell division out of control, and we can get a ton of cells as a result of this one cell being out of control. That one cell that had in its DNA, it had a SNP, which deformed, or it made the incorrect amino acid, deformed a protein, caused the signaling pathway not to work correctly, caused it to divide out of control, and then we get this clump of cells, which is a tumor. And remember, not all tumors are cancerous. You can have a benign tumor that's essentially harmless, or a malignant one that is metastatic and invasive, which means that it can spread throughout the body. But there are a lot of ifs here. So we had to get from SNP to an amino acid to protein, signaling pathway, and then cell division out of control. Not every single SNP is going to cause a changed amino acid. Not every altered amino acid is going to cause a deformed protein. And even if a protein is deformed, maybe the signaling pathway won't even be disrupted. So there are a lot of ifs. So next we're going to talk about how frequently the damaging mutations actually occur. Alright, so now let's look at one theory on how a genetic mutation in a single cell can result in an entire tumor. And once we look at that, then we can shed some more light on how frequently it actually occurs. So one theory of how this of how cancer develops is the multi-hit hypothesis. And we're going to look at this first, and once we understand how the hypothesis, what it's proposing, then we can better understand how frequently cancer can occur. So with the multi-hit hypothesis, the idea here is that there's a minimum number of mutations required to lead to a cancer cell. So each of these mutations could be described similar to what we showed in the last slide, where we have that progression all the way from a SNP to an amino acid, protein, altering maybe a signaling pathway, and then eventually altering, or causing some sort of phenotypic change. And so with the multi-hit hypothesis, there are classically 6 hits, and each of these hits is a different mutation in a particular cell's genome. So one classic hit is in the cell cycle. So normally the cell cycle has these 2 checkpoints, and what these checkpoints are for is to make sure that all the DNA is correct. So when a cell is dividing, it has to replicate its DNA before it can split into 2 daughter cells. And what can happen with cancer is maybe one of these checkpoints is just bypassed, and we just go right around it. And now all of a sudden, we're replicating without checking that the DNA is actually correct, and that can definitely be a problem later on. Another one of these hits classically causes deformation of the cell. So maybe the cell is supposed to look something like this, but it actually looks something like this. And again, that can affect the cell's ability to function properly. Another very important hit is contributes to angiogenesis. So if the cell is going to divide out of control and build this clump of cells, this tumor, then it's going to need some sort of a blood supply, right? And that's what angiogenesis means. It means growth of new blood vessels. And so if your tumor wants to grow, it has to have a new blood supply. And so one of those hits in that original cell is thought to be dealing with angiogenesis. Another one of the hits deals with telomeres, which we haven't talked about yet, but essentially they're this little tail that appears on the end of DNA when you're copying it. And basically every time that you copy DNA, it gets a little bit shorter. So maybe you copy it once, and all of a sudden that's gone. You copy it again, now we lose a little bit more. And so as you lose more and more, you can, what ends up happening, we won't talk about it in the grainy detail, but you can get problems with replication of DNA. Another problem that the cancer has to figure out a way to deal with is loss of adhesive regulation. So normally cells don't, typically they don't stick together. But in a cancer, you have a tumor where you have a clump of cells. So somehow they have to figure out a way to all stick together. And so they need to lose their adhesive regulation, which is going to involve some sort of genetic mutation. And then lastly, in order to be metastatic and then ultimately invasive, they have to figure out how to get into the bloodstream, and then equally importantly, how to get out so that they can grow in another location in the body. And that's how we get cancer. So what's the takeaway from all of this? So the takeaway is that you need multiple genetic mutations before you can actually get a cancerous cell. Just having one hit probably isn't going to do it. You're going to need multiple mutations that can do multiple things within the cell. At least that's the idea within this hypothesis. And so these hits, they can be hereditary, which means that they are passed down from your parents, or they can also be acquired. So you can acquire genetic mutations throughout your life, and that could just be a result of aging, which has to do with that, possibly telomerase activity, or even your DNA just it makes mistakes sometimes, and sometimes that's unavoidable. Even UV from sunlight that can damage your DNA. So can smoking and lots of other behaviors. So ultimately, the accumulation of some combination of these mutations can be, it can cause cancer.